Feeling any better?
A little bit.
Okay, so we had a review session last night. It's posted on the internet. It's about 2 hours, and I got to be honest, we kind of ramble on and off here and there on topic and off topic. So you can fast forward to stuff that sounds boring. Like if you don't want to know about Erin's gallbladder, then skip that section where she tells us all about having that taken out and things like that. They did, while we were studying, point out some things that we could add, or at least maybe clarify on the review sheets. So I'll fix those at some point. But like under lipids, we decided that I had skipped a part that just said what are some general properties of them. Now that's on the homework. So really, do I need to put it on the study guide? I don't know, because they're both supposed to be complementary, right? But it's worth listing. And then maybe you could list the categories of lipids. Well, it's more like we took stuff out. And I don't know why you guys are so fixated on the page numbers. If you want, I won't leave answer blanks on it, and it fits on one page.
No, I mean like the study guide. Basically, for the most part, we need to know like literally everything on that.
Yeah.
Yeah, that's 17 sheet.
See, that's what I'm saying, is the number of pages doesn't mean anything, because if you got rid of all the boxes, it would be one page of stuff you need to know.
Could fit all that on one page. Try.
Okay, that's a challenge I'll take up later.
Right, well,
So,
you have to be able to read it.
for instance, you could list the categories of lipids. For instance, there's one category we didn't even talk about in class, that's the miscellaneous category, but you should still at least know what was in that category. Under reactions, we didn't really talk about it, but on the list I forgot to mention dehydrogenation. So if we take something like this, and remember this reaction goes both ways, so I can subtract hydrogen from it, and that is our dehydrogenation to make a double bond. Or if I go the opposite way and add hydrogen to that, so I'm adding it across the double bond, then that's a hydrogenation. So hydrogenation and dehydrogenation. Meaning we put one but not the other, and that's probably just me forgetting because I think they're the same reaction. If something's the same, kind of going forward and backwards, I like to say they're the same. We should point out that one of the things that you should be kind of thinking about is some of the comparisons and contrasts. And this won't necessarily be a question on the test, contrasts, whatever, but it helps you to place the material in your brain between carbs, lipids, and amino acids. Meaning, like the polysaccharids were simply lots of carbohydrate monomers put together. Phospholipids and some of the bigger lipids are just smaller lipids put together. Proteins are just giant amino acids. There are a lot of amino acids strung together. So that, there's some similarities. Or how we connect two carbohydrates together, how we connect lipids together to make complex lipids, and how we connect amino acids together, 99% the same thing. So if you understand one, hopefully you understand all three. Or if you can make that, what? Of how they're connected together? Like all three of them or one specifically? Which one do you want? Well, let's put it this way. Here's a carbohydrate, right? And here's another part of a carbohydrate. If I want to stick those together, what type of reaction is that? Minus H2O, right? So we found the water and we realized that it just makes an ether bond. Oops. That's an ugly looking sugar, but you get the idea, plus water. If I'm making parts of a phospholipid, for instance, if I've got glycerol and a carboxylic acid, and this is not showing all of this parts because we're trying to see the part that matters and then not do the part that's repetitive, Again, what type of reaction is it? Minus H2O, and so we get, that part looking like an ester plus H2O, and we won't worry about the rest of it. Or if you look at a sphingolipid, it's the same way. And if you look at amides, they're a little bit different, but the general idea is still the same. That we're losing water in that reaction. And so if you can figure out what two parts of the molecule have to go together, and I don't know, it's harder to draw this one, and maybe I should just draw one of the NHs as not an NH2 and say that's an NH and it's got an H. Now you can see where that water comes from. And so then you've got your NH2-CH-C double bond O dash NH-CH-C double bond O OH, and you've got your different R groups hanging down off of that. So if you can realize that all three of those are the same, and you just have to know what part of the molecules or where to find the water in the molecules, it's not too bad. And then if you practice a few times for each one, so you don't make weird types of mistakes or you don't combine the wrong parts of the molecule together, then you probably should have it for the test. And it'll probably show up on the final too, because that's the most basic fundamental reactions that are involved in each of those molecules. It's the reaction that is used to make bigger and bigger and bigger molecules. Plus, it's all dehydration, so it's not too bad. And then, of course, if you're kind of thinking, you should also realize that we can always just do the reverse, which is hydrolysis. But to me, if you understand minus H2O, you should probably be able to understand plus 2H2O. They're kind of different sides of a coin. So that's what I mean by comparing and contrasting and looking for similarities in how we talk about things. The other thing that came up last night is after reading chapter 30 and understanding how enzymes function, if you go back and reread chapter 28 and 29, they make more sense. That a lot of the vocab in chapter 28 where it's talking about, a series of chemical reactions and that arachidonic acid through the action of cyclooxygenase turns into like thromboxanes and all those other types. Well, you now understand that cyclooxygenase is simply an enzyme, right? And you could even realize that when it talks about atherosclerosis and it also talks about how to treat inflammation from overexpression of the arachidonic acid pathways, that it starts talking about inhibitors and activators and feed forward kind of and feedback inhibition and sort of things like that. Meaning chapter 30 maybe should be covered before chapter 28 or maybe even chapter 27. You could probably see doing it that way. So if you go back and reread some of those chapters, you might understand more things. And then the other thing we decided that was missing from the study guide is you really should be able to draw a picture that illustrates atherosclerosis in that whole process. Atherosclerosis. I think that's how it's spelled. And that you, instead of memorizing like all the parts and how it functions, if you draw that picture, it almost answers all of the other questions. Meaning if you can sketch the diagram, you know what HDL, LDL, and VLDL, what their roles are. You know which ones are good and which ones are bad. You know the basic process by which this occurs. And so that picture might be worth 1,000 words. I might have a test question which simply says, draw a picture illustrating atherosclerosis or write a paragraph with 1,000 words. I won't, but Well, it's literally in your book and it's literally drawn in your notes. Did you want me to redraw it for you? Or can I just draw,
I know what it is, I just didn't know if there was
Oh yeah, I mean it's on page 779. And so if you can sketch that picture, to me that's how I remember the things. I can't remember for the life of me the difference between VLDL, LDL, and HDL. If I draw the picture, and sketch in the role of each, then it explains to me what each does. I can never remember which one's good or bad because it's a 50-50 thing in life. For some reason, I can remember whether omega-3s are good and bad. For some reason, I can remember that trans fats are bad because I think you see it on every darn food label you see. But I can never remember the difference between whether LDLs or HDLs are good or bad. Does anyone know off the top of their head?
HDLs are good.
Apparently you guys are smarter than me. It's one of those left-right things that I always get screwed up. So anyway, that's something that could be added to that study guide. The other thing we talked about a little bit was if you're not sure how to answer a question, write down everything that you know that might help you. Meaning, kind of play a word game with things. Like, for instance, if I asked a question, what are 5 good uses for proteins? Well, you might look and say, gosh, I didn't study 5 good uses for proteins. But start thinking about what words we've associated with proteins. What's the first word that you think of when you think of protein? There you go. What's another one? Yeah, enzyme. So if you start playing the word games, you may not have studied all 5 uses, but I'll bet you could probably come up with 4 or 5 off the top of your head by kind of associating those words. Or, you know, if the question asks about a biological pathway or something, and it asks about arachidonic acid, but you remember the one for cholesterol, write down the information for cholesterol and then say, okay, how does that relate to arachidonic acid? Because really, I would say probably the question could be phrased, you know, it's the same question, you just substitute the word arachidonic acid versus cholesterol versus, you know, another pathway. It's the same question with different words in it. And so if you can answer one question, you should be able to maybe piece together enough about the other one. So kind of play word association games with yourself if you don't have something totally learned. Other random stuff, read an interesting article. This is actually came out a couple years ago. Apparently it's been sitting in my folder for a while, but I read it because I was bored. Kind of combines what we're going to talk about in this chapter with nucleic acids and colon cancer. We're not going to specifically talk about colon cancer, but about the cancer process. But it also starts talking about how one way to combat cancer, or that this specific cancer actually shuts down a gene or an enzyme that would combat it. So it itself is an inhibitor for something that would be, you know, leading to programmed cell death. So kind of research right now in cancer is trying to figure out, okay, we know that cancer is caused because the cells don't die and they divide too fast. Well, what about a gene that would make them divide slower? Well, if we can figure out an activator for that gene, then we can do it. One of the ones they say for colon cancer, 2 of the activators are simply butyrate, and I don't know what butyrate is. I can guess it's 4 carbons, right? And it's kind of the common name for it. I don't remember if they give the real name in it somewhere. And then like niacin or vitamin B complexes. They also said that one of the reasons why high fiber diets people have less colon cancer is it turns out fiber is an activator for that biochemical pathway. So I just think it's interesting if you look at a lot of science, hopefully you can start kind of understanding what's going on. Like Jess said, she went to the hospital and they did a throat swab and talked about how they'd have to send it off and grow it or whatever. She's like, oh yeah, I know all that. I'm in microbiology. So, you know, all the stuff we're talking about hopefully helps you understand other things. So anyway, we kind of got off topic because we didn't start our new chapter, but that's okay. End goal is, if you want, go ahead and click on the 2-hour review. We mostly focused on lipids, but we kind of went off topic and went all over the place. We didn't really talk about anything in chapter 30. We started talking about chapter 29 on amino acids a little bit. At least tried to highlight some stuff. But there is a lot of material that could potentially be on this test. Maybe I should only have it over 2 chapters. I don't know. I'm kind of debating that, but we'll see how it goes. I think you guys are tough.
Also, I don't want to have to remember something from chapter.
Well, it's a tough call. So we're on chapter 31, which is nucleic acids. And this would be sort of our 4th major category of molecules, because I don't count enzymes as being separate from proteins. To me, they're kind of the same thing, just an extension of it, even though your book gives it its own chapter. And why are nucleic acids important? Yeah, they're the component, or what we think of when we say DNA and RNA. And what that really means is they're responsible for heredity. I thought it was interesting. I mean, if you think about it, it took them a long, long, long time to figure out, what is it in you that causes you to be a human versus a dog or something else. They used to think it was the proteins, that it was very specific enzymes, that humans had one set of enzymes and dogs had another set of enzymes. But it turns out that most of the enzymes between humans and dogs are pretty much the same thing. And so that it's how they're expressed, and that's contained or controlled by the heredity. We will, kind of as an overview, kind of look at structure. And this is where I consider the chemistry to occur, so you can figure that there's a good chunk of that on the test. And then we'll talk about, as a major topic, DNA replication. Although I have to be honest, you've probably gotten more of it in Bio 111 and Bio 204 than this book will ever cover. This is supposed to be a broad view of it, so it doesn't go into much depth. Same thing with RNA transcription and looking at the processes. Again, you might have gotten actually more of it in Bio 111 and Bio 204. You might find this chapter to be easy in some respects for that. And then the last category, we'll look at the synthesis of proteins. So not the structure of proteins, not how they function as enzymes or anything like that, but literally how they're built, how they're coded from the DNA all the way to actually building the protein. And then the last category is what I call the miscellaneous category. That is variable, what we end up talking about. Sometimes we get off topic and talk about one of them just by accident. Sometimes we focus on just a couple of them and say read the rest of them in the book. It's all a matter of timing and things like that. So when you're kind of thinking about the test, realize that category 1 is probably the most important, but certainly there will be questions on all of the other things. So let's take a look at structure. And maybe we should even just say, instead of DNA and things like that, we'll just start with nucleic acids. And so I kind of think about it as a process. So just like, for instance, when we looked at the structure of starch, we had to first understand glucose, then we understood how to build disaccharides, and then we learned how to build bigger molecules. Same thing with proteins. We first had to learn about the individual amino acids, then we learned how they go together to form peptides, then we looked at how they form enzymes, and then things like primary, secondary, tertiary, and quaternary structure. Same thing here. We're going to build up from the bottom part and build it into a bigger and bigger molecule. And what I'm going to primarily hold you responsible for is sort of building or being able to draw and understand and interpret those pictures. So first off, you're given the 5 bases, plus you're also given the structure of ribose and deoxyribose. Meaning you don't have to memorize the building blocks. You do have to know how to put the building blocks together. So let's take a look at sort of the basics of the 5 bases. There's 2 sort of precursor molecules that they're built on, and that is purine and pyrimidine. And so you can kind of think about purine and pyrimidine as the very most basic possible structure for each of those. So if we look at purine, and again, you do not have to memorize these structures. What I want you to do is kind of look at them critically and think, what type of reactions can these undergo? What sort of things can happen? And I certainly will never memorize these. I suppose maybe I would sooner or later, just after I teach this class enough, but I would never hold you guys responsible. So that's what purine looks like. It's a 6-membered ring and a 5-membered ring. And then pyrimidine's easier. It's just a 6-membered ring. Like that. So one of the things to note that's different is there's a large amount of nitrogen in these molecules. And remember, like things like sugars had just carbon, hydrogen, and oxygen. Lipids basically were carbon, hydrogen, oxygen, and some phosphorus, and a little bit of an amine here and there. And you start getting the DNA bases, and now we start kind of expanding it, and we notice that there's a lot of nitrogen contained in these. So these are kind of the cores. And then what happens is, from the purine molecule, you get two bases. You get adenine, and you get guanine. And probably you guys are familiar more with if we abbreviate them with their single letters for DNA, right? Does that make sense? In fact, there's years where, honestly, I forget exactly how the name is pronounced because I've just, at some point, just used all the letters. And then the pyrimidine one, there's three bases associated with that. There's cytosine, thiamine, and uracil. And that's C, T, and U. And so those abbreviations are what we'll probably end up using after a while instead of actually drawing the pictures of the molecules. But to start with, and certainly for the test, you can figure that you'll be drawing at least one example of this. The nice thing is you get this in your book and on the cheat sheet, right? So this is what's on the cheat sheet. Now there's something in common that all those molecules have. So all bases have either a 1' or 9' NH group. And what we're going to realize is that's approximately equal to the OH group that we see in sugars, that we see in lipids, and kind of like the NH, well, I guess we actually draw it this way, C double bond O, OH, to NH parts. Meaning they're the handle, they're what's going to react on these molecules. So if we take a look at an example of one of them, we'll take a look at adenine. 5-membered rings. I'll draw it kind of decent, like this, like this, like this, like this, like this, like this. Nitrogen, double bond, N-H, NH2, N, double bond N. Now again, if you want, these are on page 853 in your book. They are on your cheat sheet. You don't have to memorize them. If you want to sketch them in your notes, go ahead. A certain part of me wants to always, for this chapter, make stickers because they're a pain in the butt to draw and just be able to slap a sticker down on your notes or something. I don't know, that makes me feel like I would be in grade school then, potentially. So notice that if we number these molecules, for instance, we can go like this. 1, 2, 3, 4, 5, 6, 7, 8, 9. And we should point out that all of these are really 1', 2', 3', 4', 5', 6', 7', 8', 9'. What do you think that little prime means? What is that little dash used, or why do we have to have it? Well, the reason it turns out is that sugars are numbered 1 through 6, so that means that these bases have to be numbered like 1' through 9', meaning the primes simply mean we're talking about the base. If we're talking about just the number 1, 2, 3, 4, 5, 6, then we're talking about the, actually, do I have this backwards? Actually, I have it backwards. Never mind. 1 and 9. Wait, let me think. Which ones get the primes? Yeah, actually, this is good. We'll leave it like that. Except your book does it the opposite. Actually, your book doesn't even really focus on that. This is one of those things where it depends on the book. So this is book dependent, which one gets which, it seems. So what I really kind of want to call your attention to is simply that they're all going to have an NH group someplace on the molecule. Some of them actually have more than one, but it's the NH that's part of the ring, and you know, it's either the 1' or the 9' one. Again, you get the cheat sheet. It's almost highlighted on the cheat sheet so you can tell which one it is. So you don't even have to think very hard. Just like, you know, you're given the structure of glucose, all you have to do is figure out how to connect it together. So the first part is the bases. And then the second part that we want to kind of look at is how the bases are connected to the sugar. So we've got two sugars, ribose and deoxyribose. Again, on your cheat sheets, but the main reason we want to do them is we want to look at it. So they're 5-carbon sugars, so they make what type of ring? What do we call the 5-membered rings? Yeah, furanose rings. And so here is our D-ribose. And then the other version, the deoxyribose, is almost exactly what it sounds like, in that we simply lose one of the OH groups. So this is called 2', well, D-2' deoxyribose. And all that's talking about is that the number 2 carbon has lost its OH group and just has a hydrogen there. This is really the difference between what we think of as DNA and RNA. Whether there's an OH in one spot or not on the molecule. That's one of those things I kind of think, you know, it's like, wow, it's amazing, but maybe it's not so amazing. I mean, if ribose works pretty well for what we're doing, then it seems like if you just remove an OH group, that's a nice handy way for your body to distinguish between two different things. It's just one of those minor little changes, like the difference between alpha and beta glycosidic bonds being the difference between starch and cellulose, or you know, how simply changing a D or an L isomer for proteins can sometimes change their functionality a lot, or simply exchanging one amino acid for another in a giant enzyme can reduce its functionality by a large amount. It's just these small changes in structure that cause things. So we've got our bases and we've got our sugars, and then the next step up is what we call nucleosides. And so sometimes when I think about this, you know, I kind of like to diagram things out, so I'll be like, oh yeah, nucleosides are just a base and a sugar. They're stuck together basically. So it's just a base plus the sugar. And so how are we going to connect those together? Well, we're going to take the amine on the base plus an OH on the sugar, so really an alcohol. And what does an amine plus an alcohol make? So this is technically a secondary amine. Actually, it just makes a tertiary amine, minus H2O. So let's see, I'm going to pick an easy one to draw. Heck, my notes, I don't even actually have an example drawn. I just have an ugly picture. So what we're really saying, for instance, is if I take my sugar, and then I've got an OH here, plus a base, and we got a double bond there. So which base did I draw? See, here's what it is. I don't even know which base I drew. I literally just don't have that memorized because I don't want it. So this is cytosine. And this is ribose or deoxyribose? Yeah, ribose. because I've got the OH there. So we always react it between the one carbon on the sugar and in this case, because it's cytosine, this is the number 1 amine on that. And so here's our OH. And so then if we stick those two parts together, really you're just simply redrawing the molecule again. Nothing else. I think this gets tedious, but that's okay. You only have to usually do it once on a test. And so then that's got an oxygen, oh, no, that goes straight to the nitrogen on our molecule. And then that nitrogen is part of that 6-membered ring. And that 6-membered ring has a ketone group in it, another nitrogen, and a double bond. So if I've got the sugar and that stuck together, then this is what we really refer to as C. Or in this case, this is just cytidine. Oh, I'm sorry. Yeah, I was not paying attention. And this is kind of a crappy looking double bond there since I threw the double bonds part on the outside. And if I simply remove this, we can abbreviate it as simply DC, or deoxycytidine. Deoxycytidine. I guess I can kind of fit it in there. Again, don't have to memorize any of that. You just have to know how to put those parts together. So if you can do that chemical reaction, then you've got it. And of course, I can ask for any one of the 10 combinations, right? So don't memorize them all. Just learn which carbons and which nitrogens react together. So that is a nucleoside. Then the next step up, so the way I kind of like to think about it is a base and a sugar. Now they go together to make a nucleoside. And then if I add a phosphate group to that, now we can have our nucleotides. So a nucleotide is simply a base sugar and a PO4 group in it. And I got to be honest, 9 times out of 10 I mix up nucleosides and nucleotides because what's the only difference between them? Yeah, it's one of those just annoying little things in life. I can never remember a good way to do it. The only thing I can think of is which letter comes first, S or T? So nucleosides come before nucleotides. This is how I learn. If I can't figure out, or if two words are so close in meaning to each other, I have to come up with some stupid way to remember it. And so I remember S comes before T. So nucleosides come before nucleotides. And so what we do is we react the 2', 3', or 2, 3, or 5 OH group on ribose, or the 3 or 5 group OH group on deoxyribose, with a phosphate. And remember, depending upon how we're drawing the phosphate, a lot of times books will write it like this. Sometimes they write it as PO4 3- because these all have minuses. Sometimes you'll see them actually just write P with a little subscript I, which stands for phosphate. And really what we should do is realize that when we're doing it in a reaction, oops, I leaned on the corner and good, it didn't crash. We really actually start off with phosphoric acid, so we really have all those OH groups. So we really should think about it as H3PO4. We just realized that normally those O's groups react, or those OHs react, and that generally we lose one OH or one H plus in solution. Meaning at the body pHs that we're at, that hydrogen isn't actually attached to that. Now, why can't I have a 2' or a number 2 carbon reacting on the deoxyribose? Yeah, there's no OH. That's the big difference, right?
For the last one, is it like if there's more than one NH group, is it better which one? The last nucleoside to react with,
Yes. So if you look on the cheat sheet, the NH that reacts is always pointed down and drawn like that. So if you look in your book or on the cheat sheet, because I'm pretty sure the cheat sheet I just photocopied the picture out of the book and copy-pasted it on there. So for instance, if we want to do one of those reactions, we can just pick a base. And so here's where Jay starts getting lazy. And occasionally I'll let you guys be lazy. Oops, that's not like that. That's HOCH2. This just goes to one of the bases, right? So notice I'm just calling that base. Why? Because the reaction doesn't occur on the base, and so I could draw one of the 5, you could draw one of the 5, but as long as you know how to stick it there and practice drawing it once or twice to make sure you get it right, which, by the way, the first homework assignment has lots of practice drawing stuff. And sometimes I feel like on the previous chapters I need to make more practice on homework drawing stuff, but we'll do that. Plus HOP double bond O, SH, OH. And so depending upon which one we want, depends on sort of where we draw it, and so you can kind of pick whichever one you want. I don't know which one's the best one to stick it on. Generally the number 5, so we'll just do a minus H2O on the number 5 carbon. And so what that means is I'm losing this OH, and I'm losing an H there, right? And I ran out of room, so I guess I'm actually going to draw this going down on my notes or on the board. Oops, that's the number 5 carbon. So then I get something that looks like this. This goes to some base. And then I get my CH2 group. And then I get an O, a P, double bond O. And here's where, depending upon the book, they're going to draw it differently. This is still an OH group, right? But remember, a lot of times they draw this as O minus down here because we lose that hydrogen in solution. And so some books will show that as an OH, some books will show that as an O minus. Your book shows it as an O minus. I disagree. I think they should just draw it as an OH, simply because then your reactions actually balance, right? I won't care. If you draw it as an OH or an O minus, I won't make a difference. Now, your next, well, you won't have a next organic chemistry instructor. Your next organic chemistry instructor might tell you to draw it the exact opposite way. I don't really care. I don't think that makes a big difference. As long as you understand that the reason the H is missing is because it's lost to solution generally. And so there's our phosphate. And the way we name these molecules, oh, I see, naming plus. So we name the base. Then we give our location. Then we use mono, di, or tri, and that's really just the number of phosphates. And then we use our phosphate. So, for example, if I said the base we used was adenosine, then we would have called this molecule adenosine. Actually, since we would have, this is the, oh no, adenosine dash 5 prime dash monophosphate. Oops, monophosphate. So notice that we've got our base, a location, the number of phosphates attached, and the word phosphate. And then the key to it is to realize that if I added another phosphate on that 5' group, then we would just call that diphosphate, right? And then we can also have abbreviations. And the way the abbreviation works is we normally write it as a location, the base, the mono, di, tri, and then a P for phosphate. So we would call this 5' AMP, meaning 5' AMP simply says we have adenosine 5' monophosphate, or we basically got a phosphate group on the 5' carbon. I don't know if memorizing the rule works better or simply, seeing a name and the abbreviation written down a whole bunch of times makes more sense. So you need to be familiar with both, because when you're reading, they might say the name out and they might use the abbreviations. So a good question might be, for example, draw 3' D, I don't know, GDP. So I kind of picked randomly letters and things like that. So you have to take a look at each part, right? What does D mean? What does G mean? What does D mean? And so I always start by drawing the ribose part first. And so deoxy means that there's no OH there, right? So either specifically draw on the hydrogens like that to show it, or just leave it blank. 3' means that the one that's going to react is this one right here, right? And so that goes to, there, we'll draw it like that, an oxygen, a P, double bond O, dash O minus. And since it's diphosphate, what does that mean we have together? 2 phosphates. And because we're total slackers, I'm not even going to bother drawing the guanine up there. I'm just going to write the letter G, because I'm a slacker. I have to admit it. Well, you have to draw a couple of them on the homework and one on the test probably? Sure. So practice it, but, we've shown you an example of it, so it's just changing whatever is attached to it. If they just say like ATP, by default it's the 5' spot. So we should make a note that quite often they assume, oh my gosh, that pisses me off because they shouldn't. They assume if no number, it's the 5' spot. So ATP really is the same thing as 5' ATP, meaning we should say where it's at. So the sugar numbering, was that what you were going to ask? Yeah. Is 1, 2, 3, 4, and 5. So for instance, can you ever have a 4? No. No, because there's no OH group that'll react, right? And for deoxyribose, can you ever have a 2? No, there's no OH there to react. Hey, look, I even have in my notes a little big asterisk that says, mention that 5' is the assumed default position if it's not given. I will always tell you where it's at, because it seems kind of like a nice thing to do. Okay, so we've been going along our merry way, where we've looked at bases and sugars. We realize that they go together to make nucleosides because S is before T. And then we realize that if we add some PO4 into the mix, we get nucleotides. And then if we get nucleotides together, meaning multiple nucleotides, we get what we call nucleic acids. Or really what we're kind of saying is you get polynucleotides. I think that's a better name than nucleic acids personally. In fact, I don't know, I should ask Todd this someday. He might know the answer. I'm not sure why they call them nucleic acids anyway. I don't see a lot of acid functional groups on them, and I think it's just one of those stupid names that isn't quite right, but we get stuck with because that's what someone named it the first time. And so I think polynucleotides are better. If I kind of like to think about this, I kind of think about this as being things like starch or enzymes, meaning it's just a whole bunch of them stuck together. And back here we have our glucose and our amino acids, you know, if we're kind of thinking or trying to draw analogies between them. Now, there's a lot more parts to making nucleotides than there is to making, you know, starch and things like that. So the analogy breaks down a little bit in that there is no really nucleotide counterpart in sugars and in proteins because it's just sticking the same of a lot of things together. Yeah, we'll cover more of that later. Let's just do one last thing that we want to take a look at. We want to talk about the structure of ATP. And the reason we're kind of sticking it here is, first off, it's where your book sticks it, meaning it's one of those, you got to put it someplace. And because it really is a nucleotide, and so before we move on and start talking about polynucleotides, we should really focus on ATP. And so if we take a look at that structure, remember that's really 5', right? Is really the way it should be. And so, you know, here I've got my adenosine, so I've got an OH, CH2-O-P-O-P-. Is that 3 of them? Nope, that's only 2. Oops. Dash P dash OH and O, and O, and O. I sometimes like to think about this as a pop molecule because of all the POPs. And we'll be exceptionally lazy and just put a giant A there to tell us that that's the base that we're using. And heck, looking in your book, I see they're even lazier. They just showed the phosphates and then they didn't bother showing even the sugar part of it, but that's okay. So why do you guys know about ATP? Yeah, it's an energy storage molecule, right? Meaning if you've taken any biology, it crops up all over the place. It's how your body uses, it's one of the main routes that your body uses for transferring energy or supplying energy that is required for chemical reactions. Why is it an energy storage molecule, though? Did they ever get into that? So the energy is actually being stored in this bond here and this bond here primarily. And these are sometimes referred to as high energy phosphate bonds. Oops, don't crash. Oh, I don't want to have to draw that again. Foobar. Should have known it was going to happen sooner or later. Oh, it's good and dead. Okay, we'll just delete it. We're going to end here anyway, but I'll redraw it real quick. Maybe I won't draw the whole thing. Phosphate bonds. Actually, it's technically called high-energy phosphate anhydride bonds. Sometimes I like to leave the anhydride off just because it's a mouthful, but we'll put it on there. And now, like I said, I'll draw this here for us all out good. Again, we might take one shortcut, which is we will just focus on what we need, which is the HO-P-O-P-O-P-O. That's 3 phosphates. And then this is my A part. We're just not even going to draw the sugar this time, because what I really want to focus on is just this part right here. I should also point out that some books will draw the O minuses up and the double bonds down, and some, things like that. And that is that this bond here, oops, let's switch colors, keep it consistent. And this bond here I called high energy phosphate and hydride bonds. Now remember, what does anhydride just mean? It means just minus H2O. And so they're formed by losing that. And they contain approximately 35 kilojoules of energy per mole. And the reason that they kind of store energy, the reason that you can see that there, is it's due to the closeness of the negative charges. So we know that opposites attract and likes repel. So in order to bring those OHs close together, it requires a lot of energy. And so when we break that ATP molecule, then we can release that energy and use it for a chemical reaction. Now, why they chose ATP over a bunch of other energy storage molecules, I don't know. There probably is some reasons you could think of if you go really deep into it. But it turns out to be such a good molecule for this that it pretty much is universal. And so if we kind of think about it in terms of a reaction, we have ATP plus H2O goes to ADP plus PO4 plus 35 kilo, oops, that's a 5. Do you want me to go back a slide? And so here we use energy. Well, I guess maybe we should say we release energy. And going the opposite direction, we store energy. But it really is simply just a hydrolysis or a dehydration reaction. And remember this PO4, you might see this written as PI in some books. It just means inorganic phosphate. Or, I don't know, I guess they're too lazy to write O4, because I think writing O4 isn't too hard. Let's see. Do I have anything else we need to talk about? We'll certainly see much more of this in chapters 33 through 35, which is really bioenergetics, or how we get and extract energy from most of these molecules, or how we build them up in the body too. I think that's probably a good good spot to stop. This should be enough to do homework 31A, and so we'll just say that's due Friday. Okay, that's that.